everyone, welcome to CUET PG Lesson Session, and this is me Shalini. So in this session, we people are going to cover the concept of electrophoresis. Okay, it is a very important technique used in the molecular biology field. Okay, so the reason for using this electrophoresis technique is to purify and separate the macromolecules like DNA, protein, and RNA. Okay, so we can separate the molecules based on the charge, size, and the mass. Okay, when we are applying some sort of electrical field, the molecules will be separated. Okay, so we will see what is electrophoresis, what are the types of electrophoresis in the session. Don't miss the session, watch till end. Okay, let's move on to the session now. Okay, let's begin with the electrophoresis. Okay, so electrophoresis is nothing but when we are applying the electrical field to the sample will be migrated. Okay, so the migration will be depends upon the size, mass, and the charge. Okay, so this technique is very much helpful in separating molecules like DNA, proteins, peptide, amino acids, and the RNA molecules. Okay, so apart from the molecular biology field, it can used in the different kind of fields like forensic, for criminology, lot of techniques, like lot of fields, this electrophoresis technique will be very much helpful. Okay, so the main reason is to purify and separate the molecule based on the size and mass by applying the electrical field. Okay, so there will be two different electrode will be present. One is cathode, another one is anode. The cathode means it is negative charge and the anode will be positive charge. When we applying the electrical field, the particles will be migrated, moving from the negative charge, cathode to anode, positive charge. So, okay, so the electro means it is nothing but electricity. Phoresis means movement. Okay, by passing electricity to the sample, there will be a movement of ions. Okay, there will be a particle will be moved in the gel. Okay, so this was the first technique invented by Drusius in the year of 1807. Okay, so for the separation of the molecules, this particular technique has been invented. Okay, so how the technique is performing, how the electrophoresis technique is performing. So the principle is behind us nothing but movement of particle based on the size charge. When we applying the field, the electrical current to, okay, so the samples will be moved from negative to positive. Okay, and in order to separate the sample based on the size, charge, and the mass. Okay, it is a very important principle. Just make a note of it. Okay, so when we are ready to separate the sample of DNA, so usually the DNA will be negative charge. Due to the phosphate ions present in the structure of DNA, the DNA can be easily separated from the cathode to anode. Okay, so the electrophoresis movement only from the negative to positive side. Okay, so when we are applying the DNA, when we are ready to separate the DNA, it can be easily passed because it is already a negative ions. Okay, but if it is a protein, the protein is carrying a positive charge. Okay, so it is actually a complex 3D model structure. Okay, the protein will be in the 3-dimensional. Okay, so for applying the negative charge, the sample will be treated with the SDS, sodium dodecyl sulfate. When we are treating with this SDS, the complex structure will be break down and the negative charge will be applied throughout the primary structure. Okay, so when we breaking down the 3D dimensional structure, it will result in the primary structure. Okay, only the amino acid linkages will be there. So through the linkages, the SDS is anionic detergent, which will apply the negative charge throughout the structure. Okay, so now the protein become negative. When we going to run the sample, it can easily run from the cathode to positive side. Okay, so this how we are running the DNA as well as the protein sample. Okay, so when we are putting in the supporting medium, how the rate of migration is done. Okay, it will be depend upon the certain factor. First thing, the charge of molecule. What kind of charge does the molecule carries? For DNA, it is negative charge. For protein, it need to be changed into negative charge. Okay, and the next one is size and the shape of molecule. How if we are going for the entire genomic study, so we need to break down the DNA. Okay, so we need to use the restriction enzymes for break down the DNA. Okay, so the size and the shape is depends. Okay, so if a DNA is very large or if it is with a high mass, it will be deposited at the starting itself. Okay, it does not run throughout the gel. So if it is very small, it will run throughout the gel. It will form a ladder. Okay, so size and the shape of molecule is very much important. And then next one is electrical field. How we are passing the electrical field. So depends upon the sample, the electrical current will be different. Okay, and if we give over current, the sample will run fast and it will be able to mix up with that buffer also. Okay, so strength of the electrical field is important. And the next one is supporting medium. So for DNA, we will use agarose as a medium. For protein, we will use a stacking gel and the resolving gel, which composition will be different from the agarose. So the supporting medium for the sample is very much important. Depends upon the sample, we will select the supporting medium. And the next one is temperature. So how the entire system is going to be maintained. Okay. So the rate of migration will be depends upon the certain factors like net charge of the molecule, size and shape, strength, supporting medium, and the temperature. Okay. So this is about the principle of electrophoresis. The next we will move on to the types. So there are a lot of types like gel agarose, gel electrophoresis, STS stage, 2D dimensional electrophoresis, pulse field. Apart from this, capillary, immunoelectrophoresis, there are multiples of types have been evolved. Okay, but the basic thing we are mainly concentrating on the agarose and the SDS page. Okay, so the common step for every electrophoresis is first thing isolation of sample. So for running the electrophoresis technique, we need to isolate the sample. Either it can be a protein or it can be a DNA. Okay, or the RNA sample can be also used for the separation. And the next thing is gel preparation. So for DNA, we will select agarose gel. For protein, we will select a resolving and stacking gel. Okay, we will see how the gel has been prepared in the later slides. Okay, for gel, the supporting medium is very much needed for carrying the sample. The next thing is casting. So it is like a tray, a glass tray where the gel will be poured and it will well has to be set for carrying the sample. Okay. And the next thing, after casting, the electrode is going to be connected with the casting. There are two electrodes. One is cathode, another one is anode. Okay. So the cathode, it is negative. Anode, it will be a positive charge. After the gel run, after the sample run, we can observe under the UV illuminator. The sample has to be observed under the UV illuminator or the gel down. Okay, so it will reflect how many bands has been formed, how the sample's weight has been, what kind of weight is there. So we can easily observe with the UV illuminator. Okay, so these are the steps common for all the electrophoresis. Little bit it will be changes for separate kind of electrophoresis. We will see that. Okay. Next, so the first thing will be isolation. So either we can isolate the DNA sample, RNA sample, protein sample, or the peptide, whatever. So depends upon the need of sample, we can isolate. So for example, I'm giving the DNA extraction. The DNA can be taken from lot of sources. It can be a blood or any kind of fluid. Even in the plant extract, banana extract, onion extract, we can make the DNA. Okay, so the DNA, even the bacteria. So let us take a bacteria cell culture. The cell is extracted and the cell is sliced. When the cell breaks, it will release protein, DNA, and the RNA. Okay, it will release the all compounds in the environment. Now we have to treat the sample with protease because we need only DNA, right? So we need to remove the proteins. So with the help of protease, the protein is removed. Okay, after that, we are going to treat it with RNAs. Okay, now the RNA will also eliminate. RNAs will destroy the RNA nature. Protease will destroy the protein. Okay, so finally the sample will have only the DNA. Okay, it is purified, and for maintaining the pH, for maintaining for the long period, the TE buffer will be added to this purified DNA. Okay, now the sample has been ready. Let's move on to the electrophoresis steps. Okay, so it will involve multiples of steps. First thing will be sample preparation. We have already seen how the DNA is isolated. Okay, so after isolated, we have to add a bromophenol blue, which is an indicator. Okay, so usually the sample is colorless. When we are putting in the well, the sample will be run. Okay, we cannot visualize where it is and where it is running. Okay, for the visualization process, the bromophenol dye has to be added. After that, the ETBR, ethylene bromide, it is a carcinogenic substance. It will attach on the DNA phosphate group. Okay, so why we are adding this ETBR is to observe when we observe under the UV illuminator. When after the sample running, when we observe under the UV illuminator, the sample has to show the fluoride. It has a capacity of fluorating. Okay, so the only if a DNA is present at the below and the ladder was formed, the ETBR will give orange color. Okay, so for the reflection, this ETBR is going to be added. Okay, first step, DNA along with the bromophenol dye and the ETBR is added. The next thing is agarose preparation. It is nothing but it is a polysaccharide taken from the seaweed, extracted from the seaweed. So for forming a pores, for preparing a small pores to which can allow the samples to run, the agarose is ready. Okay, so 1 gram of agarose is added with 100 ml of TAE buffer. Okay, it is a buffer to maintain the pH, to maintain the state of supporting medium. This TAE buffer is used. Okay, so when we applying the electrical field during the process, this buffer will help to separate the sample. Okay, and the agarose will be 1 gram and along with that 100 ml TAE buffer will be used. Okay, so this agarose will form a cross linkage and which will allow the sample to move. Okay, for protein, we will not use the agarose, but that the medium will be different. Okay, there will be two different kind of medium, stacking gel and the resolving gel, and then we will use like acrylamide, bisacrylamide. The different composition will be used for the protein separation. Okay, so now we have seen sample, and the next one is supporting medium separation, so preparation. Okay, the next thing will be gel casting. So after the agarose is prepared, it has to be poured in a tray. So you can see in this diagram. So the glass plate is nothing but casting tray. We have to cover the two sides, and then the prepared agarose, the 1% of agarose which we prepared, we are going to pour in the casting tray. Okay, and allow them to solidify. Okay, when we are heating, the gel will be in the liquid form. After cooling process, it will be in the solidified form. Okay, so before the solidification, we have to place the comb. You can see the third diagram. So the comb was inserted in the agarose gel for forming a well. Okay, so in that well only we are going to load the DNA sample. After the well is formed, now the DNA sample has to be inserted. Okay, the concentration of DNA has to be selected and has to be inserted. So for sample, we have taken bromophenol blue, ETBR and the DNA. Now with the help of micropipette, the DNA has to be loaded in the well. Okay, now after loading, there will be two electrode, cathode and anode, that will be connected. Cathode means negative. The negative charge will have a black color wire. For anode, it will have a red color. Okay, so the wire has to be connected. Now the voltage has to be increased. Now while after the electrical field supply, the molecules will start moving. Okay, the molecule will start separating. If it is higher molecular weight, the molecules will deposit near the well itself. Okay, if it is very small molecular weight, it will move throughout the gel. Okay, you can see it in the diagram. After the separation, it will be looking like a ladder. Okay, so the third step is casting, which is where the tray has to be sealed and the agarose gel will be poured. Okay, and after before like little bit solidification, the comb has to be placed for preparing the well. Okay, so the next step will be the well is loaded with a sample, DNA sample, which we are isolated. After the loaded, the electrodes is to be connected and the electric field has to be pushed where the sample will be separated. Okay, it is looking like a ladder. This is how the agarose gel electrophoresis is work. Okay, so next we will move on to the SDS page. So SDS nothing but sodium dodecyl sulfide polyacrylamide gel electrophoresis. Okay, so this SDS page is mainly used to run the protein sample, protein related sample based on the molecular weight. We have to separate the proteins. Okay, for that purpose, we will use the SDS page. Okay, so it is a very important technique in the molecular biology as well as the biochemistry field. Okay, so like other electrophoresis, this SDS page is also working based on the principle of when we passing the electrical field, the molecules will be separated based on the charge, size, and the mass. Okay, so this is about the SDS page. And what are the chemicals we are going to use in the SDS page? The first thing is sodium dodecyl sulfate. As I already told, it is an anionic detergent. in order to denaturate the protein structure, in order to break down the complex structure of protein, and it will make the primary structure. Okay, so because it need to apply the negative charge, the electrophoresis run from the negative to positive charge. Okay, so the protein is actually a positive one. So when we are using the sodium dodecyl sulfate, the structure will be break and it become a primary structure, and throughout the structure, the negative charges will be applied. Okay, so that it can easily move. Next chemical will be beta mercaptoethanol. So just note down, these chemicals are very important. Okay, the functions of the every chemical will be different. Okay, the next thing will be beta mercaptoethanol. So in order to break down the disulfide bond, this beta mercaptoethanol is very much important. Okay, some kind of amino acid like cysteine, methionine will have the sulfur group in their structure. Okay, it will connect with the sulfide bonds. For breaking the disulfide bond, this beta mercaptoethanol is important. Okay, so the sample need to be added with sodium dodecyl sulfate as well as the beta mercaptoethanol. Okay, and the next, the supporting medium will be acrylamide and the bisaccharidine. So both are the gel matrix. When we mix up both, it will form a cross linkage. It will form a pores. Okay, so the acrylamide will be acting as a backbone and the bisacrylamide acting as a cross linkage. Okay, when we form a like a net, it will form a like a net so that the sample can be easily run. Okay, so the next one will be tris HCl. So this is a running buffer which will helpful to for the movement of sample. Okay, and this kind of buffer will maintain the pH of the entire system. Okay, and the next thing will be ammonium persulfate and termite. Okay, so these two are the chemicals which will helpful for the formation of cross linkage. Okay, it will be acting as a catalyst and it will allow the bisacrylamide and acrylamide to form like a polymerase. Okay, it will allow for the polymerization process. And the next thing is glycerol. So glycerol is added to sample because when we adding in the well, the sample has to be deposited in the well for the movement. If it is floating, it will mix up with the buffer, running buffer. Okay, so for giving the density to the sample, the glycerol will be added to the sample. Okay, and the last thing will be composite blue, brilliant blue. So we have seen one dye in dagros, right, for bromophenol blue. So for visualization, we are using that. So likewise, for visualization of protein bonds, this composite brilliant blue dye is used. Okay, so what are the chemicals required for this SDS page? Sodium dodecyl sulfate, beta mercaptoethanol, acrylamide, bisacrylamide, glycerol, tris HCl, ammonium persulfate, temide, and composite brilliant blue. Okay, so these are the chemicals which is required for running the SDS page. And the steps is also different. Okay, so for preparation of gel, in the agarose gel electrophoresis, only agarose is a medium. But for SDS, here two medium has to be prepared. One is resolving gel and the stacking gel. Both gel are made with the acrylamide, bisacrylamide, buffer solution, ammonium persulfate, and temide. But the concentration of these chemicals will be different. Okay, so the resolving gel will have a certain concentration of these compounds and the stacking gel will have a certain concentration. Okay, the first, after preparing these two gel, the casting tray has to be sealed over. Okay, so you can see the diagram. So this is the casting glass plate. Okay, the plate has to be sealed two sides. After sealing, the first thing, stacking gel has to be poured. The below surface, there will be a stacking gel, and the above surface, there will be a resolving gel. So after pouring the resolving gel, we should insert the comb in the gel for making the wells. Okay, so in that well only the sample is going to be run. You can see in the diagram, the below dark blue is nothing but it is a stacking gel. And the above green color is resolving gel. In the resolving gel, there will be a well formation which is made by the comb. Okay, so now after set some of the time, the sample is going to be added. The sample need to be mixed with beta mercaptoethanol, SDS, and glycerol. For density, we are adding glycerol. For SDS is to give the negative charge and beta mercaptoethanol is to break down the sulfate bonds. Okay, so apart from this, there will be a load dye is going to be added for the visualization. Okay, once the sample is loaded, the negative charge is up connected to the negative place and the positive is going to be connected with the positive. Negative means cathode, positive means anode. After connecting, the voltage is given, electrical field is performing and the sample will be run based on the charge, size and the mass. Okay, it will be separated. Okay, after that we can use chromazine brilliant proof for the visualization process. Okay, so after running the SDS page or after running the DNA, so the sample will be looking like this. Okay, we can compare the sample. So the marker is nothing but already known molecular weight DNA ladder. Okay, so we can easily compare. If we take our 3 unknown samples and along with that unknown sample, when we run the marker, we can easily compare that this unknown sample weight is similar to this marker. Okay, so the left side there is a marker. There it is also represented in the ladder form, which is known molecular weight. Okay, and that ABC samples are nothing but unknown, which we have. So while we running the sample, we will get a ladder. Okay, so we can compare with the marker for identifying the molecular weight. Okay, so this how the SDS or the agros will be looking like. Okay. And we have seen SDS and the agros. Next, we are moving to the pulse speed electrophoresis. Okay, as I already told you, there are a lot of techniques of electrophoresis have been evolved. If we are small, we have small sample of DNA, we can go for agros electrophoresis. If it is more than, if we want to study the entire genome, okay, entire DNA in a living system, we can prefer the pulse field electrophoresis. Okay, so the entire genome has to be isolated. For the DNA isolation, we have seen the steps. What are the first thing? Protease has to be treated. After that, RNAs will be treated, and finally, we will get a DNA purified form. That DNA will be digested with the restriction enzyme. So the restriction enzyme is nothing but it can identify the site where the DNA can be cleaved. Okay, it will make the large molecule of DNA into the smaller one. Okay, it will make into the fragments. After fragments, we can go for the gel running process. Okay, so pulse speed electrophoresis is nothing but there will be a complete isolation of genome. The entire genome has to be isolated and has to be digested with the restriction endonuclease enzyme. Okay, it will identify the palindromic sequence, which is like, for example, Malayalam. When we read from the both the direction, the arrangement will be more similar. Okay, so it will identify such kind of sequence and it will just cleave, it will make the fragments. Okay, so after cleavage, there will be a lot of fragments will be present in the genomic DNA. So, okay, and now after digestion, we have to prepare the agarose gel electrophoresis. Okay, now the large set of DNA has become very smaller. Okay, so now we are going for the agarose gel electrophoresis. First thing, we need to prepare sample already prepared, and the next thing we have to add the sample with bromophenol dye and ETBR. Okay, so after preparing sample, the gel has to be casted. The agarose gel need to be prepared. 1 gram of agarose along with that 100 ml of TAE buffer is added. So this is supporting medium. That supporting medium will be poured in the casting tray, and after casting, the well has to be created with a comb. So now the digested digested DNA sample will be loaded in the wells. Once loaded, the electrodes are connected, and by passing the electrical field, the samples is going to be run. Okay, so we can visualize the sample using the UV illuminator. Okay, or the gel doc is commercially available. We can use the gel doc. In the computer result, the image of the cell gel. Okay, so this is a finalized image. So this how the even when we are running the pulse speed electrophoresis, the images will be looking like this. Okay, so even in the computer, we can observe the images. Okay. Next, we will move on to the 2D electrophoresis. Okay, it is a two-dimensional gel electrophoresis. It is a very powerful technique for separating a protein molecules. This electrophoretic technique is very much important. Okay, so the separation of protein will be based on two different factors. One is isoelectric point, another one is molecular weight. Okay, so the normal SDS page is to separate the molecule based on the molecular weight, size, and the charge. Okay, but first thing we have to separate the sample based on the isoelectric point. So the pH, the protein will have the normally 0 isoelectric point. Okay, the pA value of the protein will be 0. First thing we need to, this is nothing but there are two different steps. One is first dimension, another one is second dimension. The first dimension step is isolation of protein based on the isoelectric point. And the next thing is second dimension, based on the molecular weight, we are going to separate the protein. Okay, so there was a strip called IEF, isoelectric strip, which is coated with a gel. When we are inserting in the protein sample, the sample has to be extracted. The sample can be whatever. Okay, even from the plants also we can separate the protein. After protein separation, the IEF, that isoelectric gel coating strip, has to be inserted in the protein sample. Okay, and it will allow to run. After separation, you can see the diagram. Okay, the protein extract, they have inserted a strip called IEF, which is coated with gel. Okay, based on the isoelectric point of pH, the colors, that color represents the protein separation. Okay, the protein has to be separated. After that, up to this, the first dimension is over. Next step will be SDS page. So next we have to separate based on the molecular weight. Okay, so the SDS page, we will have two different gel. One is stacking gel, another one is resolving gel. Okay, so stacking gel is prepared with the bisacrylamide, acrylamide, timide, ammonium persulfate, so on. So likewise, with a different concentration, the resolving gel also prepared. After pouring the gel in the casting tray, instead of we need to place this IEF, that strip, okay, which is already separated based on isoelectric point. Okay, so we have to place the strip instead of well. Okay, after placing, the electrode has to be connected and run for the sometimes. Okay, so now the sample will be separated based on the mass. Okay, once again I'm telling, so 2D electrophoresis, it will be like a very important technique for the separation of protein based on two things. One is isoelectric point, another one is molecular weight. Okay, so different kind of protein will have a different kind of isoelectric point. Usually it will be 0. So first thing, the protein sample has to be extracted. After extraction, the IEF strip, which is gel coated strip, will be inserted into the protein. So, and allow it to separate. So the color represented in the IEF strip is the protein separation. So after that, now we have separated based on the isoelectric part. Next, we will move on to the molecular weight separation. For that, the SDS page is prepared. So with the two different gel, stacking gel, another one is resolving gel. Okay, so in the resolving gel, we will make a well, right? So instead of well, there will be we have to replace the IEF strip in that well. Okay, after that, the electrode will be connected and the samples will be separated based on the size and molecular weight of protein. Okay, so there will be two different one is isoelectric, another one is molecular weight. Okay, so this is about the 2D electrophoresis technique. Okay, apart from that, there are capillary electrophoresis, immunoelectrophoresis, lot of things. Okay, so in this session, we have covered agarose, SDS page, and pulse field and 2D dimensional electrophoresis. Okay, so I'll give the recap. So the electrophoresis is nothing but it is a separation technique. We can separate a protein, DNA samples. Okay, by passing the electrical field, the molecules will be separated based on the size, charge, and mass. Okay, so the sample can run from only negative charge to positive charge. Okay, so the DNA, when we apply applying in the electrophoretic field, it can easily move. Because it carries a DNA, which carries a negative charge. If it is protein, it carries a positive charge that has to be made to negative charge with the help of SDS and beta mercaptoethanol. After that, the gel will be run with the medium called agarose medium and the stacking gel medium, polyacrylamide gel medium. If it is protein, we will go for the polyacrylamide gel medium. For DNA, it is agarose medium. Okay. So for for DNA based sample, we will use gel electrophoresis, agarose gel electrophoresis. If it is small sample, we can go for agarose gel electrophoresis. The agarose will be the medium. If it is we have to study the entire set of DNA, we can prefer the pulse field electrophoresis. Okay, that DNA has to be digested, and after making into fragments, we will move for the separation technique. Okay, again, that pulse field will also move back to the agarose gel. Okay, for protein separation, it is SDS page. sodium dodecyl polyacrylamide gel electrophoresis. The medium will be polyacrylamide gel. Okay. So this is about the electrophoresis. Hope you have understand the concept. Meet you in the next session. Thank you.